Two pictures of a scientist. It's probably the same person, one called a geographer, one called the astronomer. This is the one of them. It's actually in the Louvre. And I like to think this could be Leeuwenhoek. But I have to admit to you, there is no good evidence for this speculation. Although when Vermeer died, which he did in his 40s, very early, which is why he has only limited numbers of paintings that survived, Leeuwenhoek was the executor of his will. So almost certainly they did know each other. Well, over the next 2 centuries, it became clear that all living things, all living organisms, consist either of a single cell or a collection of cells. This conclusion was nicely summed up by this rather serious German scientist of the early 19th century, Theodor Schwann, who wrote in German, this is a translation of course, "We have seen that all organisms are composed of essentially like parts, namely of cells." This meant, of course, that the cell is the basic structural unit of life. But as I've already mentioned, they are also the basic functional unit of life. And what do I mean by that? Well, it was described by another German scientist, Rudolf Virchow, who actually founded the medical science of pathology. And he expressed it well with this quote here from 1858. "Every animal appears as a sum of vital units, living units, each of which bears in itself the complete characteristics of life." In other words, cells express the characteristics of life, the functions of life. It's really actually quite important. He also said that all cells come from cells. He wasn't the first to say that. He said it in Latin, omnis cellula est cellula, which means cells arise through the division of pre-existing cells. I'm going to refer back to that at the end of my talk, but that dealt with those who thought that cells could just arise spontaneously out of mud or murky pools. So, cells are interesting. Now, I hope I've convinced you of that, but if I haven't, I'm going to show you another picture. Look at this picture. This is a picture of a mammalian egg being fertilized by sperm. And I need to remind all of you that you all once looked exactly like this. All of you were once a single cell. And if it's not interesting for you, there is some curtains there on a door, though I think it's been locked now, so it's probably best to leave now, okay? But this is really, I think it's quite profound. We were all once a single cell. Now, cells are considered to be the simplest entities which unambiguously exhibit the characteristics of life. I say unambiguously because I will have a few words to say about viruses and the like, which sort of strike a sort of intermediate existence. And this means, of course, that understanding cells is key to understanding life. It's not key to understanding everything about life, like how brains work and so on, but it is key to understanding what we mean by something that is living. And that means cells figure prominently in what I have to say. This is a cell, a modern EM cell, artificially colored. They are bounded physical entities. They're separated from their environment, but they're surrounded by a semi-permeable lipid membrane. Semi-permeable because it can control what can come into the cell and what can go out of the cell. So what this does is it separates a cell from its environmental surroundings, but is in communication with those surroundings. Why have I emphasized that? Well, I emphasize it because this separation allows order to be built up inside cells at the expense of increasing disorder outside cells. And I want to emphasize that because in this way, life does not contravene the second law of thermodynamics, beloved by physicists who often get troubled with life, which seems to be going into the opposite direction of what that law suggests. It certainly troubled Schrödinger as a physicist, and he, a couple of quite nice sentences here. The first is, an organism's, this is from the What is Life book I said at the beginning, an organism's astonishing gift of controlling a stream of order, that is permanent order, on itself, and thus escaping the decay into atomic chaos. That's what troubled him. He saw everything decaying into atomic chaos, but life persisted, and persisted for generation after generation. I've taken a second quote from that book. These chromosomes, he was talking about chromosomes, I'll say some more about those in a few minutes, contain in some kind of code script the entire pattern of the individual's future development and functioning. And it was this code script which Schrödinger, I think, thought allowed life to escape the decay into atomic chaos, because that code script was being inherited from one life form to another. And this quote, of course, introduces my second idea, and that is the gene, which explains the mysterious phenomenon of inheritance. This idea, the second I'm going to talk to you about, the gene as the basis of heredity, had its origins with the work of an Augustinian monk, Gregor Mendel. Mendel was abbot of Brno Monastery, which I visited actually. I helped them set up a museum for him. And he published two extraordinary papers in 1866. Here he is, Gregor Mendel. And he tackled the problem of inheritance by carrying out crosses of pea plants with different characteristics and analyzing the outcomes in thousands of the progeny formed in these crosses. I found this picture of his garden in 1981, when I was living in Gladsmoor Road, actually. When I visited the monastery, this is what it looked like. In 1981, it was the middle of the Cold War, of course, at that time. But I want you to look at this. This is a monastery. That garden, which was his research garden, was huge, if you think about it there. And it had a greenhouse of the same size. Adjacent to it, which since, by the time this photograph had decayed, although they've now replaced it. And what struck me was the monastery was like a little research institute. We don't think of monasteries as research institutes, but actually it was full of teaching monks, of which Mendel was one, and they did research. He was a physicist by training. He studied the weather, he did a map of the moon and things of this sort, but he spent a number of years working on plants and plant breeding. And what Mendel did, as I've mentioned, is crossed peas of different characteristics, plants which had different characteristics, different heights, you see here, different flower colors, different seed shapes and so on. But he was very methodical about it. I think took a sort of physicist's clarity of thinking, not just descriptive, but counting. That was really critical for him. So he had characteristics he could clearly distinguish. He did crosses, and he didn't simply describe what types came out of those crosses, he counted the types. And he famously observed, and you probably learnt about these things even at school, he famously observed simple ratios in the characteristics of the progeny produced by these crosses. Ratios like 3 to 1 and 9 to 3 to 3 to 1 and so on, and 1 to 2 to 1. And this eventually led to the idea of Mendelism. That sentence was written very carefully because he didn't quite get to Mendelism. Although Mendelism is what he's known for, he almost got there. It had to be refined about 30 years later. And this is the idea that heredity is based on pairs of indivisible particles he called them factors, one of which is inherited from one germ cell, say an egg, another which is inherited by the other one, say a sperm, or in the case of a plant, pollen and an ovule. And these factors were coming together, and it was the interaction between these two factors which generated what the progeny looked like. Now, nobody took any notice of what Mendel did until about 30 years later, when chromosomes were discovered, because these factors were subsequently found, about 50 years after he did this work, to be linked to the chromosomes. And I'm going to show you, I think I have a picture here of a 19th century chromosomes. Do you see those sort of strings? This is actually an onion tip squash. The first time I looked at cells was under a microscope out in northwest London, and saw exactly what you see here. These chromosomes separated every time the cell divided. So scientists thinking about inheritance thought, well, the characteristics could be inherited on these chromosomes because they're nicely separating out every time a cell divides. These Mendelian hereditary factors, I think, were much more acceptable when they were seen under a microscope. Mendel's thinking was rather abstract, especially for biologists of the time. But when that could be translated into a concrete observation, that is the one we're seeing here, then it became real. Because abstraction is always a bit abstract, and this gave you reality. Now, we now call these factors genes, and in the 1940s, Oswald Avery in Rockefeller University, where I was president for a while, in New York, showed that genes were made of deoxyribonucleic acid, or DNA, published it in 1944. And then the structure of it, and here's the structure, was shown to be a double helix established 10 years later here in the UK, in London by Rosalind Franklin, Ryan Gosling and Maurice Wilkins, in Cambridge, interpreted by Francis Crick and Jim Watson. And you see here the chromosome being unraveled to produce what is the iconic double helical structure. And Crick and Watson famously argued that the double helix structure of DNA could explain heredity through the linear sequence of bases making up the DNA and the pairing between complementary bases, which I'll explain in a moment, where one side of a chain can pair with the other chain through two molecules, adenine pairing with thymine, A with T, cytosine with guanine, C with G. This is a double helix, and imagine this as a ladder with rungs. Now twist it. The twist just gets in the way, to be quite honest. I mean, nobody thinks of the twist. But if you imagine it a ladder, you now separate the ladder so the rungs are split. If you have G always pairing with C and A always with T, then you now have a template that you can precisely copy. And this was a huge insight. And it can explain how DNA is copied during cell reproduction, ultimately accounting for heredity. It explains Schrödinger's code script and the permanence of the genes between generations. And because DNA is a code script, which is what was proposed, and I'll explain in a moment, then that's precisely copied every time a cell divides. Crick went on to propose what he called the central dogma. He liked words like this. I got to know him quite well. I would never call something a central dogma, I have to say, but he was a very competent person. Okay, and this explained how the DNA encodes the structure of proteins, which we're going to talk about in a moment. The central dogma describes how the sequence of bases making up a gene is copied from DNA to RNA, ribonucleic acids. That acts as a messenger between the DNA located in the nucleus, as shown over here, where the chromosomes are, and the cytoplasm, where proteins are made. And there is, to a first approximation, one gene encoding each protein. These were fantastic discoveries in our lifetime, and they lay because I think we're all of a similar age, in our lifetime, and laid the foundations for molecular biology and are critical for understanding life. Central to this is this genetic code, written in 4 letters, the 4 bases I've described to you, with the information, I want to emphasize this, stored in a linear form. This way of storing information is, in fact, the same as the sentences that I'm talking to you, or the sentences you read in a book. They are linear encoded information. It's also similar, of course, to bytes in a computer, which are exactly the same. And what it means is that the linear digital storage of information was invented by life over 3 billion years before the computer age. Over 3 billion years ago. We've just discovered it. Well, we discovered it with language, but in the last 20,000 years. But basically, it was discovered by life. So the nature and function of the gene introduces the 2 next ideas, which I want to tell you about, sometimes slightly technical in places. So if you doze off, I'm okay with that. I'll pull you back a bit later, okay? The first is this as life as chemistry, and the second one is as life as information, looking much more holistically at the problem. So the first one here, how chemical activity and physical forces bring about life. Now, the origins of this idea, notice, by the way, I started with the cell, 17th century. We got to genes, which was 19th century. We did get into the 20th century with the nature of genes, though. But the idea that life is based on chemistry can be traced back to Lavoisier, the father of chemistry, who started some speculations in the late 18th century, unfortunately lost his head in the French Revolution, which terminated his research, of course. And there's a very famous painting by David in the Metropolitan in New York. And I discovered, and it was really shocking, really, that David was on, 10 years after he painted it, was on the jury that condemned him to death 10 years later. Extraordinary. Well, at least I found it extraordinary. This idea can be traced back to him, but it was Louis Pasteur, another French chemist, who carried out experiments on sugar beet fermentation in the mid-19th century, which led to this concept. What Pasteur was doing, and this is a romantic picture of Pasteur, I think it's in the Louvre, might be in the Pasteur Institute, actually, I can't remember. Pasteur was analysing, forgive me, was analysing the fermentation of sugar beet to make alcohol at the request of industrialists in northern France, because the fermentations that they were doing with the sugar beet to make alcohol failed sometimes and made acid instead. And he demonstrated that successful production of alcohol required the activity of a single-cell living organism, yeast. And he proposed that the yeast cell produced substances that made alcohol from the sugar, of the sugar beet. And if the yeast was not there, but bacteria instead, the fermentation made acid. So he said to the industrialist, put yeast in and it'll always work. Okay? But he concluded more generally the following, that fermentation was a physiological act yielding products for the cell, that chemical reactions are an expression of the life of the cell. This is direct translation of what he said. In other words, life is chemistry. But I tell you, it's a very elaborate and very special chemistry. It's based on carbon polymers, linear structures, DNA and RNA are those. And now we need to look at carbon polymers that make up proteins, because these are the substances from yeast that Pasteur postulated made the alcohol. Proteins are polymers of amino acids with a backbone and side chains of amino acids. The backbone is the bottom line here, made up of carbon, C, and nitrogen, M. And then each of these are fundamentally the same unit, but they have different side chains, lysine, leucine, tyrosine, aspartic acid, and those different side chains have different chemical properties. Lysine is very basic. For example, aspartic acid is acidic, leucine doesn't like water, and so on. Tyrosine is very bulky. And what this means is that, that because there's 20 of these different amino acids, that as a result of this, there's a wide variety of chemistry that can be carried out by proteins. And this contrasts with DNA, which chemically is very stable and very boring. Very stable, but really boring. But the chains of amino acids making, chemically, the chains of amino acids making up protein polymers it can fold in different ways. This is just actually looking, before we leave it, what the structure looks like. But this is a chain of amino acids, and because they chemically interact with each other, they can make different sorts of shapes, and therefore order the acidic residues, the positive residues, the ones that don't like water, in particular places inside the protein. And you can make grooves where different sorts of chemistry can take place. And this results in molecular structure like this. So what this is doing is turning a linear structure into a three-dimensional chemical structure capable of undergoing chemical work. So enzymes and other proteins essentially act as chemical machines. And these machines produce a huge range of different chemicals for living organisms to function. They make new molecules, they break them down, they recycle them, they make big molecular assemblies, they make nucleic acids and other proteins and so on. But these chemical machines can also do physical things. They can do physical work. They can make motors which carry cargoes around the cell on trackways, like sort of little railways. They act in hybrid ways, combining chemistry and physics, capturing energy from the sun to produce energy-rich chemicals that can drive the activities of other chemical reactions. And all of this amazing, and it is truly amazing, amazing diversity of chemistry is going on simultaneously and close to all sorts of other chemical reactions in cells that are 10 micrometers across. And thousands of reactions going on in a wide variety of different conditions in exactly close proximity. I put up here a map of metabolism I had to learn when I was an undergraduate many years ago. For those of you who remember the Krebs cycle, that's the red thing in the middle there, for example. This has got some hundreds of reactions, but there's thousands of reactions that are going on. Now, what I want to explain to you is these reactions only work if they are working in slightly different sorts of conditions, because they're specific for the different chemistries. And that means they have to be somehow separated one from another. And that can be separated by the folds in the protein, or it can be separated by putting complexes of proteins together, or by having, within cells, membrane-bounded what are called organelles, like the nucleus or the mitochondrion, which separate the chemistries one from another. It's not the way this is normally described, but it's the way I want you to think about it. Look back to this cell. It isn't just a static structure. It's actually a highly dynamic set of many, many different chemical microenvironments. And these allow the hugely elaborate chemistry of life to take place. But for all of this to work as a whole, these chemical reactions have got to be connected. You see the sort of the problem we have here. They have to be separate, but if they can't talk to each other, there is no chance of getting it to work for the overall function of the cell, to have purpose. I'm deliberately using the word purpose because it agitates people a bit in these circumstances. Anyway, but so they have to communicate one to another. Has to be organized. Has to be communication. And communication requires the transfer of information. And that leads to the next idea, life as information, which is also central to understanding life. And central to this is life is a complex system. It consists of lots of controls, and it generates purposeful behaviors of the whole. I tried to find out where this was first discussed. Quite often you read books where people say this has all just been invented to think of it like this. That isn't true. It isn't always thought about like this. And I think you'll be surprised to find that I discovered 25 pages of this in a book written by Immanuel Kant around 1800. And he was very interested in life as a complex system. I was astonished. I think it was on moral philosophy, so goodness knows what it was doing there. Now, the operations of complex systems are based on the management of information, and I'm going to give you two examples to illustrate this. The first, I've already explained to you, so I wanted to use this, because the structure of DNA is iconic and beautiful, although I have to say, when I set up the Crick Institute, I said, I do not want a piece of art that looks like a double helix anywhere near it, okay? But it only makes sense biologically when you recognize it, as I've already explained, as a digital information storage device. That's that's the way it makes biological sense. One written in this linear script of 4 letters. Now, a linear polymer of DNA is ideal for storing information. The encoding letter bases are turned inside, protected from chemical change, and there is no chemical activity going on, quite different from proteins. But, and this is the clever thing about life, this information can be turned into chemical action that can do work through those varied chemical activities of the proteins, which are encoded by the genes. And for me, this is a wonderful characteristic of life, which is not emphasized enough. It's all built on these carbon-based polymers, the ability to encode information in inert, one-dimensional linear structures, such as DNA, and yet convert that into three-dimensional, chemically diverse structures, which are the protein, which can carry out the chemical and physical work. This is astounding. This is why life works. It's really critical. Second example I want to tell you about is about regulation. That is making complex systems in living organisms to act as a whole. A cell grows and divides into 2. That's why I call it purposeful. That is what it's actually achieving. I don't mean purpose in the sense of, catching the bus, but I do mean that it is purposeful as acting as a whole. This is a familiar concept, more easily understood in man-made machines. This is the governor developed by James Watt to regulate steam engines. And I don't know if you're like me, I understand most machines built before 1900 and increasingly none after that time. And this one we all understand. It's driving a steam engine, the spindle spinning. It spins out these balls that lifts up a valve and shots off steam. So the engine slows down and they go back in again. Very easy to understand. Look at an integrated circuit and try and understand that. I gave up years ago. This is, in fact, an example of a negative feedback loop up here. Back now to the chemistry of life. We've got a molecule A going to a molecule B going to a molecule C. C inhibits A to B. So as C builds up, C switches off A to B, and so C can drop. Negative feedback loop responsible for homeostasis, very simply. Positive feedback, complete opposite. As A goes to B to C, as C builds up, it accelerates A to B. So once you've triggered this, it's a switch. You go from one state of no C to another state of lots of C. And it's an irreversible switch. Many such feedbacks and control modules operate in cells. They maintain homeostasis, they generate switches, timers, toggles, oscillators, translating chemistry into informational modules. And these can be linked together to generate even more elaborate informational management. As a metaphor for this I quite like, which is the electronic circuit that most of us don't quite get our heads around, where you have lots of components. By the way, some wit, I stole this from a biological journal, and the proteins were put here to show they're all linked together in this complex system sort of way. But actually, a better metaphor is not hardware, which is what this is, but wetware, because living cells operate in solution chemistry. And that means you can relink things through solution chemistry. So it's much more versatile than our computers. Now, the management of life's chemistry by information allows, as I've said, for the cell to have overall purpose. Reproducing the cell, I mentioned, making a butterfly, maintaining our cells. Now, such purposeful behavior was thought to require a designer or a divine creator, for example. But it took evolution by natural selection to suggest there was an alternative. And that's the final idea I want to explain to you. And then we're going to have a go at putting all of this together. This is evolution by natural selection, how living things come about. Now, this is probably the most beautiful idea in biology. We identify it mainly, quite rightly, with Charles Darwin. The idea has two parts, though. One is that life evolves over time, aeons of time, but over time. And secondly, a major mechanism for that is natural selection. That's the two ideas. Now, this is a picture of Charles Darwin over there. And this was his father, Erasmus Darwin. And I'm going to talk a little bit about Erasmus Darwin, because the fact that life evolves over time was not Charles's idea. It was an idea talked about quite a lot 50 to 100 years before him, particularly by Jean-Baptiste Lamarck and by Charles's eccentric grandfather, Erasmus Darwin. Erasmus Darwin was one of the founders of the Lunar Society in the Midlands that met once a month under the full moon. I think they were nicknamed the Lunatics. They came together and probably drank too much, but had interesting ideas, scientists, engineers, and so on. Erasmus was colourful. He was a scientist, a doctor, and a poet. He wrote all his research up in the form of poems. I kid you not. I quite like him, and I bought most of his first edition, and it's all written in poetry. So if he wanted to talk about plant taxonomy, he wrote a book called The Loves of Plants, and it's all written there in blank verse. He was a Republican. He refused to be George III's physician, for example. He was a proponent of women's education and wrote a little textbook on that. And he was a great conversationalist in the Midlands, nowhere near any university, by the way, in Derby and Lichfield, despite having an appalling stammer. Very interesting person. And he and others like Lamarck thought there was a tree of life, that is, that living organisms are related. However, they had no idea about the mechanism of it. Now, if life only arose and survived once, and that is probably likely given that the chemistry and coding found in all living things from bacteria to our cells is the same, so we can't be sure of that, but it seems likely, then that argues that all life on Earth is related to each other. We are related to everything on this planet. I'm going to return to that at the end a little bit. This connection of life was captured in the only diagram in Darwin's Origin of Species, published in 1859, where he illustrated the first tree of life. you'll see all these sort of branches emerging and some going extinct. So at the top line you have the survivors. Notice at the bottom, A to L, he wasn't sure whether life arose once or many times, and he sat on the fence over that. He didn't know anything about the chemistry, of course. He gathered evidence in favour of this, because this had been speculated before, when he was the naturalist on the Beagle, the ship that did surveying for 5 years around the world. And there he developed his idea of natural selection initially, or just after he came back. So what is natural selection? It's the consequence of populations of a species exhibiting variations caused by inheritable genetic changes. So it's related to the things I've already been talking about. These genetic changes can be caused when there are mistakes, when the DNA is copied, or it's damaged by external causes. Now, if these variants influence characteristics that make organisms more successful, better adapted, and therefore reproduce more, and that's the key thing, because it's related to life, then that particular genetic variation will spread through the population. And over time, such genetic variations will accumulate and lead to changes in the population. Such individuals of that new population eventually will no longer be able to breed with the original population. That's natural selection in a few sentences, but it may be a bit short. This can lead to a new species which is better adapted and more successful in survival and reproduction. He came to this view in part by his study of finches, the beaks of finches on Galapagos Islands. I didn't draw this myself. I stole it from somebody else. I forget where. Scientific American, I think. Darwin's finches are here. Basically, they're adapted to do different things. They came from a single finch. The Galapagos are volcanic islands, so they haven't been there for very long. The one at the top breaks nuts. You can see that. The one at the bottom catches insects under bark. These have occurred by natural selection. On the right, we have a set of pliers, okay, all clearly related, but designed intelligently designed by human beings for a particular purpose of doing different things in, electricians and so on. But the main message here, of course, is one is occurred by natural selection, the other by intelligent design. And, of course, natural selection doesn't require a creator, whereas the one on the right does. Now, for natural selection to take place, you need three things. Reproduction, of course, a hereditary system, the gene, and that hereditary system needs to exhibit variability upon which selection can work. And that, you'll note, is based on the ideas of the cell and the gene. So they're all linked, these ideas, which I haven't emphasized enough. I'll give you one simple example. Imagine a single-celled life form. It's got a brown cell coat, the color of which is determined by a gene. Imagine that gene is not copied properly. Now it makes a red coat. Imagine that red-coated cells survive better. Maybe it avoids predators, doesn't matter what. As a consequence, the red cells replace the brown cells in the population, and natural selection has taken place. I mean, just linking the ideas, and you see it here. It's a very important idea in biology, so much so that the 20th century geneticist Hermann Müller used it to define life. He defined life as living things have properties which allow them to undergo natural selection and to evolve. I'm going to return to that in a moment, and if you're getting worried this is going on all night, it's only a few more minutes, okay? So a deep breath, soon be done, okay? But before I sum up, I want to say goodbye to Darwin as an old man and read out his final monumental sentence from his Origin of Species, whilst this planet has gone cycling on according to the fixed law of gravity from so simple a beginning. Endless forms, most beautiful and most wonderful, have been and are being evolved. Why can't we write scientific papers like this today? I can't bear to read what I've written a year afterwards. I mean, this is immortal, you know. You could read it and it's terrible. By the time you've submitted something, it's gone through the editors. They've taken out anything of interesting because it's too speculative. I tell you, this sentence would not survive in a modern journal of science, I have to tell you. Now, I want you to notice how he refers to gravity. Here he's relating to the great physicist, of course, Isaac Newton, that biology can have fundamental laws, just like physics. He didn't say it because he was subtle, but that's what he meant in saying this. Right, before I sum up, let's have a brief discussion about viruses. We all know about viruses now, of course, and I'm still suffering from one at this very moment. Viruses have a genome. I think I've got a picture here. This is a bacterial virus, a bacteriophage. The genome is based on DNA or RNA. They undergo evolution by natural selection, as we saw with the evolution of the COVID virus. They thus pass Muller's test that I mentioned, but they can only reproduce when they are inside the cells of other host living organisms. And they do that by hijacking the cell's molecular machinery to copy the viral's genome and make more viruses. This means that a virus is completely dependent upon another living entity. You could also sort of say a virus is sort of alive in a cell because it does stuff, but is a lifeless chemical outside a cell. You could sort of think about that. But the question is, are viruses truly alive because of what I've just told you? Now, in thinking about this, it's important to remember that other life forms, including ourselves, are also, to a greater or lesser extent, dependent on other living things. And this is what I think is often missed out in this discussion. The obvious ones, the many parasites that live on or inside cells or bodies of animals, plants, or fungi are examples of this. But there are other examples. Other organisms are used as food. We eat plants, or we eat animals. There are certain amino acids we cannot make and are made by microbes that live within us. So what this means is there's a spectrum of living organisms. Different life forms have varied dependencies on other life forms, but nearly all of them, maybe all of them, are part of an interdependency, meaning they have dependency on other living organisms. In the case of the virus, that dependency is very strong, whilst in other living organisms, the dependency is weak. But I argue that we should think about viruses in the context of dependencies, and then it ceases to be quite such a mysterious thing. And I argue that we should consider them alive because they evolved by natural selection. But we'll come to that. But what this means is that life on Earth is fundamentally connected, both through being related, tree of life, and also through deep relationships and interdependencies. Right, I'm going to pull all of this together, and I'm going to finish with 4 slides and then say a few words after that, which summarize where I think we've got to, but I warn you, it does not lead to a neat dictionary-like definition of life. 1. Principles. Living things are independent, bounded physical entities. This allows me to get rid of computer games, by the way. They are separated from their environment, but in communication with their environment. This allows them to generate order and organization. The basic unit of life on Earth is the cell. Multicellular organisms are aggregates of cells. Next, living things possess, this is Muller's definition, life forms are entities that can undergo evolution by natural selection, By doing so, they acquire purpose. They are entities that build, maintain, and reproduce themselves, and they can evolve because they have the following attributes. They reproduce, they have a hereditary system, and it exhibits variability. 3, living things are chemical, physical, and informational machines, all three. They construct their own metabolism, didn't talk much about that, and they use this metabolism to do what I just said, maintain themselves, grow, and reproduce. These are the basic principles. I'm going to add a bit more. Life on Earth is based on carbon polymer chemistry. Nucleic acid makes linear information storage devices. Proteins are chemical and physical machines. Integration of all of this involves the management of information. And all of this means that they are connected with inside cells. Finally, living things are related by, on Earth, are related by common descent and are closely interconnected and dependent upon other living things. Now, should we ever encounter life elsewhere in the universe, and of course we do look for it, I suspect it has to be built on similar fundamental principles. The chemical details may be different. You know, maybe the science fiction writers are right. It could be based on silicon or something, depending on the environment or in which this is happening. However, to satisfy the need for long-term information storage, and it is likely, I think, that chemistry will be built on polymers, and you have to have a mechanism of transferring that information into molecules that can do chemical work. And the beauty in life as we know it is it's separated into DNA and into protein. RNA, by the way, can do a little bit of both, which is quite interesting. However, just to return to our planet, this is the only corner of the universe where we know for certain life exists. The life we are part of here on Earth surprises us constantly, but in spite of its bewildering diversity, we are making some sense of it, and that I've tried to explain to you. And it has the great potential to improve the lot of humankind and also of the natural world. But our knowledge of this is important for another reason. It's important because biology shows us that all the living organisms we know of are related and closely interact with each other. We are all bound by a deep connectedness to all other life, to bacteria, fermenting yeast, pea plants, and flitting yellow butterflies. All of which, of course, have accompanied us during this talk. All these species are life's great survivors, the latest descendants on the top there of a single, immeasurably vast family lineage that stretches back through an unbroken chain of cell divisions into the far reaches of deep time. And that I briefly mentioned at the beginning, because all of this is connected by successive cell divisions. And you can go back and back and back. And when you start thinking about that, there is a sense of wonder about it. We are connected through billions of years to everything on the planet and to organisms that existed billions of years ago. However, as far as we know, we are the only life forms who can see this deep connectivity that we know of, and in addition reflect on what it might mean. And I argue it gives us human beings a rather special responsibility for all life on this planet, made up as it is by our relatives, some close, some very distant. We need to care about it, we need to care for it, and to do that we need to understand it, which is why I've given this talk and also written a book. Thank you very much for listening to me.
Paul, thank you so much for a fantastic lecture. I think it really brings out to me the beauty of science, which I think many people don't understand, but also I think in your final words, the huge importance of this to the continuation of life on Earth, really. So thank you very much. We're going to have a few questions. Can I just speak now to the people who are watching on Zoom, because I think we've now realized that we haven't been very helpful to you, even though you can't ask questions. So to implore everybody asking a question in the room to speak right into the microphone like I'm doing, so they can hear it at home, and to keep your questions very, very short. Thank you.
Thank you, that was brilliant. You talked about Charles Darwin not being sure whether life was one event originally or may have evolved lots of times. What's your view of that?
Yeah, I think the origin of life is a deep mystery. It occurred fairly rapidly. I mean, cooling of the planet in the birth of the solar system, 4.6 billion years it formed, probably only cooled enough 4.14 billion years. We have our first fossils 3.5, 3.7 billion years, 300, 500 million years. Not such a long time to go from the building blocks of life to a functioning organism. I think it could have arisen more than once. I don't really know how that could have come about. I declare my ignorance. But what I think is likely that only one variant of it survived, which is why we had the same chemistry. So I'm putting up the white flag. I don't know how it happened. And I am saying I think it was only once, which didn't mean there wasn't more, but they didn't survive.
As you have said, we're cousins of all living things on this planet because we have a common great-great-grandfather, a single cell or whatever. To all life and all animals, we're cousins. What I've been thinking recently about the inevitability of life. If physical laws pertain all over the universe and they're all the same, given the right conditions as have appeared in this planet, and given the fact that there are maybe 2 trillion galaxies, each with 100 million stars, like our if they're similar to our galaxy, life must have appeared. Is inevitable wherever the right conditions occur, and given so many millions and millions of stars and millions of planets, this life must exist over the whole universe, maybe millions of times.
Yes, so this is the argument, because the universe is so vast. The accident, let's call it an accident that formed life, almost certainly from the probability arguments you just explained, must have occurred elsewhere. So that is certainly what that's certainly what is argued. Whether that life survived itself in the long run is a different question. Yeah.
Sorry, we can't. Can you? For the people at home, without the microphone, it's very difficult. Are there any other questions?
Over here?
Yes.
As we move forward, we're examining Mars quite intently. I suspect we might find some primitive life on Mars. Of course, the major difference between primitive life is you mentioned organelles, and of course the most important one to me is mitochondria. Do you think that's absolutely necessary to the continuation of life?
Firstly, I think we're more likely to find life on moons of Jupiter and Saturn. That's, I think, and indeed, there's spacecraft trying to collect water exploding from some of these moons. Mitochondria are the organelles that make the energy. They're found in all eukaryotes. They're not found in prokaryotes, so you can have life without mitochondria. You can make energy without mitochondria, but it is the most efficient way to do it. And all life that you can see has got mitochondria.
We're just going to have 2 more questions because it's 9 o'clock already.
Thank you for the lecture. It's really interesting. Tell me what happens with cancer. So you've talked about the building blocks of life. So what goes wrong when cancer strikes?
Yeah, okay, so cancer I know a little bit about, and cancer is actually an example of evolution by natural selection occurring in the microcosm of the body, because its ultimate explanation is in terms of genetic changes to the DNA that results in a disturbance of the controls over cell reproduction, such that cells reproduce out of control, and because they reproduce out of control, they start to dominate the organ or the tissue or the body in which they're found, and then they can get more aggressive, because the more aggressive they become, the more they will appear. So in a strange way, in the last 30 years, studies that were actually confined to sort of natural history and evolution of populations are now being applied to cancerous cells. And in our lifetimes, we knew nothing about cancer, really, 70 years ago. But in our lifetimes, we've seen a real advance in understanding and knowing which genes we've got to be thinking of, 300 or 400 of them. It's very complex. The understanding is needed. To be able to turn that understanding into practical interventions will be a long slog, but it will happen.
Okay, the last question.
Do we know much about how the genes determine morphology? For example, the pattern of dots on the butterfly wing?
Well, I tend not to use the word how genes do it, because that is a property of the whole system of how you produce an organism. But actually, it's also a property of a cell, because it has morphology and structure too. But the genes produce these chemical and physical machines that interact one with another as a consequence of information and its management, and can produce the sorts of structures that you're talking about. But probably the explanations of that are best sought in thinking about the overall system there, rather than the individual genes, because it is if you like, emerges from the interactions of lots of different components. I myself study cell reproduction. I'm interested in the generation of form and also the organization in time. I'm a geneticist, so I use genes, but I try and understand it in terms of the system as a whole.
Well, thank you very much. We do have, it's got to go at 9:15, so can I invite you all to go next door and have a glass of wine and try to nab him with your next question. We're not going to be selling books, but I'm donating my copy to the library here so that people can, over time, borrow it. It is actually a really good read and very, very interesting and accessible. So thank you.
Thank you for listening.
This
Thank you.
Good. Thank you so much.